Welcome to Metagenomic Analysis reveals a marked divergence in the structure of below-ground microbial communities at elevated CO2, a study conducted by He et al. And presented by Vanessa Ferreras. Starting with the table of contents, we will go through an introduction, the hypothesis, materials and methods, results, and the conclusion. Section 1, Introduction. This study was published in 2010 and conducted at Biocon Experiment Site in Cedar Creek Ecosystem Reserve, Minnesota, USA. The samples studied had 10 years of field exposure. Why was this study conducted? Carbon dioxide levels are rising as a consequence of fossil fuel combustion and land use changes. Effects of elevated carbon dioxide levels on plant growth and primary productivity are well established. However, robust predictions of future atmospheric conditions are hampered by uncertainties regarding the response of the biosphere to elevated carbon dioxide. Therefore, these researchers wanted to know how will elevated carbon dioxide levels impact microbial, especially below-ground communities. Some key terms used throughout the study are ECO2 for elevated carbon dioxide, ACO2 for ambient CO2, below ground, defining microbial communities within the soil as imaged, and biomass, which is the organic material from plants, animals, and waste that can be converted into energy. Researchers of this study had stated two hypotheses. 1. Microbial community composition and structure will be altered because of both the inputs of sea to soil and altered chemistry of those inputs under ECO2 and 2. Various microbial functional groups will respond differently to ECO2. In particular, genes related to C and N fixation and labile C degradation will be increased. Section 3, Materials and Methods. At the site of the study, the facility had 296 plots and 3 treatments. The first was CO2 treatments with ambient CO2 at 368 micromoles per liter and elevated at 560 micromoles per liter. Then there was nitrogen treatments, with ambient versus 4 grams N per square meter per year. And lastly, plant diversity treatment, with 1, 4, 9, or 16 species. The samples studied were taken from 24 plots with 16 species, 12 from ACO2, 12 from ECO2, and with ambient N supply. The samples analyzed were collected in 2005 and 2007 methods: DNA extraction, purification, and quantification. Soil DNA was extracted by freeze-grinding mechanical lysis. DNA quality was assessed by the ratios of 260 nanometers over 280 nanometers and 260 over 230 nanometers, and final DNA concentrations were quantified with a picogreen method, which is a fluorescent assay used to quantify double-stranded DNA. GeoChip analysis. 2 versions of GeoChips were used: GeoChip 2.0 for 22 samples taken in 2005 and GeoChip 3.0 for 24 samples taken in 2007. Lastly, 454 pyrosequencing analysis. PCR amplicons targeting V4 to V5 hypervariable regions of the 16S rRNA was performed with the 454 FLX systems with a sample tagging approach. The researchers believed it was best to combine all 3 methods to get the best results. Statistical analysis. Pre-processed data were further analyzed with different statistical methods: 1. Microbial diversity index and response ratio. 2. DCA of microbial community structure and composition. 3. ANOSIM, ADONIS, and MRPP analysis of differences of microbial communities. 4. Mantle test and canonical correspondence analysis, CCA, for linking the functional structure of microbial communities to plant or soil variables. 5. Partial mantle test and partial CCA for covariation analysis of soil and plant variables. Section 4. Results. Key results. Long-term exposure to ECO2 alters microbial community structure and potential function. Microbial and bacterial biomass significantly increase with ECO2. Genes involved in carbon and nitrogen fixation were enriched. Genes involved in labile carbon degradation were more abundant. These next 2 graphs illustrate these findings. Figure 1 shows how PFLA increased in ECO2 which can contribute to higher biomass. Excluding fungi, there is an overall trend of increased biomass from ECO2. Figure 3 demonstrates how various metabolic activities increase with ECO2. Section 5. Conclusions. Was the hypothesis proven? Yes, the study proved long-term ECO2 exposure alters microbial community composition/structure, and genes related to C and N fixation and label C degradation increased. Limitations. Some notable limitations were that the study was unable to compare the effects of abrupt and gradual increase of CO2. Additionally, it does not account for other factors associated with climate change such as temperature or precipitation. Why is this study significant? A rise in CO2 levels rise from industrial pollution, and environmental changes could result in significant microbial changes. It is unknown whether these changes will be harmful or beneficial. That concludes the discussion of this study. Thanks.